Okay, so let's go into the concept of homeostasis. Thank you so much. Let's go into the concept of homeostasis. We said that we have a green region, and this green region represents our homeostasis. Our green region represents green region represents homeostasis or the U range, and we have the U range for different variables. We have U range for temperature, thermia, that is known as euthermia. If the temperature goes up from euthermia and increases from 37 degrees Celsius up to 40 degrees Celsius, 45 degrees Celsius, what is that called? Hyperthermia. Some students say hyper. Hyper is just a prefix. There's hyperglycemia, hyperthermia, hypertension, hypercalcemia. So you have to say both parts. Hyper what? So this is hyperthermia. And if the temperature drops from from euthermia, homeostatic range, 37 degrees Celsius, down to 25 or 20 degrees Celsius is now too cold. What is that now called? So some of you still say hypo. That's the prefix. We need both. It's hypothermia. Hypothermia. Okay, so now let's look at how our body repairs. How does our body repair hypothermia and hyperthermia? Let's begin with hyperthermia. So in hyperthermia, we are up here. What happens is there's always a template. The template is there is a sensor. There is a sensor. The sensor senses that there is a deviation, either an upward deviation, hyper, or a downward deviation, hypo. So the sensor here, what is it going to be? And hypothermia. If we went up, if we are now at 45 degrees Celsius, that is going to detect hyperthermia, increase in temperature, plus, elevation of temperature, hyperthermia. Well, what kind of sensors do we have? We have mechanoreceptors, and we have thermoreceptors, and we also have chemoreceptors. Now, mechanoreceptors sense stretch. Where do we have mechanoreceptors? In the heart. So when the blood is coming out from the heart into the aorta, which is the biggest vessel, this blood, if so much blood is coming out, there will be stretch, excessive stretch of this aorta. So that stretch will be detected by mechanoreceptors or thermoreceptors or chemoreceptors. Yeah, mechanoreceptors. They're gonna detect this stretch. So what are they gonna do? They're gonna sense and they're gonna send information to the brain, and the brain is gonna say this too much stretch needs to be brought down, and then the amount of the pumping of the heart muscle is gonna get slower. The contractility will drop. The electrical stimulation to contract a lot is gonna stop. The contractility will drop. Cardiac output will drop. Heart rate will drop. So it is the mechanoreceptor that helps us here. What about when the baby is born? When the baby is pushing the cervix of the mom with the head. So when the baby is pushing the cervix, what do you think is there? Do you think there's stretch receptors who sense the stretch of the cervix? Or do you think there are mechanoreceptors? Or do you think there are chemoreceptors? What do you think? Speculate? Do you think that when the baby is pushing and stretching the cervix of the mom, who's gonna be the sensor? Is it gonna be stretch receptors or chemoreceptors or chemoreceptors, mechanoreceptors or stretch receptors? So mechano is the same thing as stretch. Chemoreceptors who detect increase in sodium level in the blood. It's like they taste blood, they taste the soup. There's so much sugar in the blood that needs to come down. We have hyperglycemia, too much sugar, or hypoglycemia, severe drop in blood sugar concentration, hypoglycemia. We don't want hypoglycemia or hyperglycemia. What do we want? What do we call it? Euglycemia. Euglycemia. You know, I wrote it here. We don't want hyperthermia or hypothermia. We want euthermia. We don't want hyperglycemia or hypoglycemia. We want euglycemia. We do not want hypercalcemia or hypocalcemia. We want eucalcemia. We don't want hypertension or hypotension. We want eutension. So now the baby pushing in the cervix. When the baby pushes, the stretch receptors, they send electrical stimulation to the brain, and the brain says the baby is pushing against the cervix, so it will secrete a hormone known as oxytocin, and the hormone oxytocin will make the uterus push and expel the baby out. So the baby's head, the baby's head is pushing against the cervix, and the baby is already, this is the uterus, and this is the cervix. So the baby pushes against the cervix, stretch receptors sense that stretch, and they send information to your hypothalamus. The hypothalamus releases oxytocin, and oxytocin causes pushing of the baby out. So here we see the baby pushes and the uterus pushes out some more. The baby push and the uterus push out some more until the baby comes out. Is this an example of negative feedback or positive feedback? Positive feedback. So the more the baby pushes, the more the uterus pushes back. There's another example of positive feedback, and that is breast milk. So when the baby suckles the breast milk from the nipple, what happens? There are stretch receptors. There are mechanoreceptors in the nipple. So these mechanoreceptors, they are sensors. They sense the baby is suckling the milk. So they send information to the brain, and the brain secretes the same hormone again, but the same hormone now is causing the milk duct to produce milk. So in the cervix, the more the baby pushed, the more the baby is pushed out. In the nipple, the more the baby suckles milk, the more milk ejected from the milk duct. Is that another example of positive feedback or negative feedback? Raise your hand if you think that's negative feedback. Positive feedback. The more the baby pushes, the more the uterus pushes the baby out. The more the baby suckles the nipple for breast milk, the more milk is ejected from the milk duct. That's a perfect example of positive feedback. There are small situations where we use positive feedback in the body. I mentioned two: in labor, giving birth, and in breast milk. The third one is when we're bleeding. When we are bleeding, let's say when we are bleeding, these are blood vessels. Blood vessels, they are not just tubes. They are made up of cells known as endothelial cells. These dots represent nucleus. Endothelial cells, endothelial cell, endothelial cell, endothelial cells. These are endothelial cells. If one endothelial cell is damaged, blood is going to start leaking out. So now we're going to have hypovolemia. Blood volume is going to drop. So we're not going to have homeostasis of blood. We're supposed to have euvolemia. Now we're going to have hypovolemia or hypervolemia. When we're losing blood, bleeding, hemorrhage, gunshot wound, blood is leaving the body. Is that an example of hypervolemia or hypovolemia? Hypovolemia. So now a sensor will sense that there is a drop, that there is a loss of blood, and then platelets are gonna start aggregating. So what's gonna happen is platelet number one gonna stick here, then platelet number two will stick on top of it, then platelet number three will stick on top of it, then platelet number 4 will stick on the top, then platelet number 5. So here we saw one platelet comes, more platelet come. Is that an example of negative feedback or positive feedback? That's positive feedback. The overall process to help us not lose blood and move from euvolemia to hypovolemia, that's homeostasis. But how did we get to the homeostasis? It was through the not a negative feedback, positive feedback. So one platelet attaches like Band-Aid. It's called platelet plug. That's why they call it platelet plug formation. Platelet plug formation. And the platelet plug formation is important to make like a band-aid, to make a band-aid to stop the loss, of blood. Is that point clear? Okay. Can you guys on that side see everything here? So then move here so you can see. Okay. All right. Is everybody able to see now? Okay. So now these are 3 examples of positive feedback. There is usefulness of the positive feedback. But do we use it for homeostasis? Do we use positive feedback for homeostasis? Do we use positive feedback for homeostasis? No. Imagine things go, we don't use negative feedback. Things go up, we go even more up. Can we ever get to homeostasis? No. So we don't use positive feedback for homeostasis because positive feedback is going to make things even worse. It's going to make is going to exacerbate the condition. So now we're going to move from hyperglycemia to even more hyperglycemia. I gave you an example of a patient who comes into the emergency room with so much very high blood glucose. Instead of giving them insulin to drop blood glucose, by mistake you give them glucagon. What's that going to do to the sugar level? Make it even more or less? Glucagon is going to add more sugar. So now we're going to move from hyperglycemia grade 1 to hyperglycemia grade 2 and 3. And that's going to lead the patient to have a coma. That was an example of a positive feedback or a negative feedback. Positive feedback. So we never use positive feedback for homeostasis because it's going to make things worse. A patient comes in with a drop in glucose. Do they need insulin that will make glucose level go down even more? Or do they need now glucagon that will make glucose level go up? Yeah, so they need glucagon. But if you make a mistake and give them insulin, and that will drop blood glucose even more, that will be an example of positive feedback. And positive feedback is going to harm the patient. So we never use positive feedback for homeostasis. There are special times where we use positive feedback when the baby is trying to eject and get out of the mother's womb, when the milk is being ejected in breast milk, and when the platelet plug formation, when we're trying to make a thrombus. So a thrombus is many platelets attached and they make a band-aid known as thrombus. And then when we stop the leakage of blood, we stop generation of these platelets. Okay, so the point is we use for homeostasis, we exclusively use negative feedback. We never use positive feedback. Very good. So we said that there is number 1. What's number 1? Number 1 is always a sensor. The sensor is going to send input. It's going to send input sensory signal, and that is going to be step number 2. It's going to send it to the computer, to the integration center. And the integration center is going to integrate this information. The integration center is going to integrate and understand that there is a deviation and we must initiate negative feedback to repair it. Then after integration is step number what? Step number 3. Step number 4 will be, so the integration center receives an input. That means it must generate a output. It's going to be an output signal. But if the input signal was sensory, the output signal must be motor. It's going to send an output motor signal to who? That's step number 4. That's step number 4. Then step number 5 is going to be, we're going to stimulate an effector, and this effector is step number 5. Then step number 6, the effector is going to help us achieve homeostasis, and that is green. Now we have achieved homeostasis. That's what we need. And after we achieve homeostasis, we must stop, and terminate the response, termination. So homeostasis, step number 6, and then stop or termination, that is step number 7. This is the template for homeostasis. If we want to repair homeostasis, we have to follow these 7 steps. Step number 1, what is step number 1? Step number 1 is a sensor. Then step number 2 is an input signal. Then step number 3 is an integration computer. Then step number 4 is a motor signal. Step number 5 is an effector. Step number 6, we return back to green homeostasis. And step number 4, we terminate the response. Okay? All right. Then, let's begin with one form of stress. So far, we know that we repair and we restore homeostasis using negative feedback or positive feedback. Negative feedback. So let's begin with when we're too hot. We said that we have a standard temperature, and the standard temperature happens to be 37 degrees Celsius. Let's begin with an increase. So now when you are too hot, 40 to 45 degrees Celsius, what's going to happen? Number 1, what's step number 1? The sensor. The sensor is going to detect what? There is a hyperthermia or hypothermia? Hyperthermia. Who is the sensor? Is it a mechanoreceptor 1 or a thermoreceptor 2 or a chemoreceptor 3? It's a thermoreceptor. Who said 3? Chemoreceptor is when you detect chemicals like calcium, sugar. The sugar is too much. Sugar is a chemical. So when you detect that chemicals are up or down, we use chemoreceptors. When we detect stretch of blood vessels for blood pressure, or stretch of the cervix, that's mechanoreceptor or a stretch receptor. But when we detect heat, that is detected by thermoreceptor. When we detect pain, there is a separate type of receptors for pain known as nociceptors. These are receptors for pain. But now we're talking about temperature, hyperthermia. We're too hot. The sun is out and we are too hot. So the number 1, which receptor do we say? It was thermoreceptor. Thermoreceptor is going to sense an increase in temperature, and the increase in temperature is going to send an input sensory signal. The input sensory signal will say what? Will say that help, there is hyperthermia. We don't want hyperthermia. That's stress. If you go up hyper something, that's stress. If you go down hypo, that's stress. We don't want either stress. We don't want hyper anything or hypo anything. We want you. So the input sensory signal is going to say that there is hyperthermia. Who is going to be the integrator here in this particular variable for temperature? Is your hypothalamus. Does that mean for every single variable out there, hypertension, glycemia, is always going to be the same integrating center? No. You know how in basketball they have different players. They have but they have the same position. They got to be a guard. They got to be a center. They got to be a forward. In soccer they have a goalkeeper. But is the same player playing in both teams? Do you have the same player playing in both teams? No. So here you're not going to have the same integrator for two different variables. The same integrator for temperature and for blood pressure and for sometimes you can have the same integrator but different locations. Like the hypothalamus has multiple nuclei. So one nuclei is responsible for the ejection of oxytocin to help the baby come out of the womb and the milk being ejected. But for the most part, we have different systems to repair different stimulus and to repair different variables and restore homeostasis. Is that point clear? So now let's keep going. So now we sense there's an increase in temperature. Then an input sensory signal was sent to the integrator who happens to be the hypothalamus here. The hypothalamus. The hypothalamus is in the brain. Then the hypothalamus will send an output motor signal to two effectors. The first effector is going to be your blood vessels. Your blood vessels. So the blood vessels now, if your blood vessels were of this size, do you think they're gonna increase in diameter like this, or do you think they're gonna get even more tiny? Or they're gonna get even more tiny? Well, they gonna when they get bigger when they get more wider, what do we call that? Vasodilation, or do we call that vasoconstriction when we dilate? When we increase the radius? Good. Vaso, because we're dealing with vessels, so we call it vasodilation. And when we decrease the blood vessel, we call that vasoconstriction. We call that vasoconstriction. Now, what helps us? We talked earlier today about temperature, about heat gradient. So now when you're so hot, who's more? So now which one is slab A? So your body is now hyper. We said it's 40 to 45 degrees Celsius. Your body is too hot. The air is warmer. The air is warmer. So what's going to happen is your hypothalamus is going to vasodilate, and when it vasodilates, it's going to vasodilate the blood vessels closer to your skin. Your skin. So it's going to be close to your skin. Above your skin, who's there? Air. So air is cooler, cool air. But your blood is hot blood. So what's going to happen? Do you think that the hot temperature is going to move from slab A, which is your blood, into the cool air? Or do you think the temperature is going to go from the cool air into your hot blood? You are hot. You have hyperthermia. You are so hot. And the air is cool. We said the two slabs, when you connect them together, the 500 degrees Celsius really hot slab, when you put it next to the 100, the heat is going to flow from the 500 really hot to the 100 until they are equal, right? So here the same thing. You're so hot, you're 45 degrees Celsius. The weather is cool. Your weather is 25 degrees Celsius. Where is the movement of heat going to be from? Is it going to go from your hot blood to the cool air, or from the cool air to your hot blood? Okay, raise your hand if you think the movement of heat is going to be from your hot blood, 45, to the cool air, 25. Raise your hand if you think that's the answer. Raise your hand if you think the heat is going to go from the cool air into your hot blood. It's going to go from your hot. Remember we said the slab was 500 and the other one, A, was 500 degrees Celsius. So hot. And B was only 100. That's still hot, but not as hot as 500. We said what's going to happen is we're going to flow from an area of a higher concentration to an area of a lower concentration. So now we will end up with this. A and B, both of them 100 plus 500, that's 600. They're going to even out at 300 and 300. But initially, where was the direction of movement? We moved from an area of a higher to an area of a lower, area of a higher heat to area of a lower heat, area of a higher temperature to area of lower temperature, area of a higher voltage electrical to an area of a lower voltage, area of more glucose, 500 glucose to area of 100 glucose until they equilibrate. So here, where is more hot? Is 45 more hot than 25, or is 25 more hot than 45? 45. So the heat movement is going to be from where? From 45 to, so now you're going to dissipate heat. Heat is leaving your body. So now when heat leaves the body, is the 45 going to go up, or is it going to drop down to 40? It's going to drop down to 40. Then it's going to drop down to 35. And then it's the heat is leaving. Heat is leaving. And then it's 25 until you hit 37 degrees Celsius. Then you have achieved homeostasis. We began with 45, so hot. And now we vasodilate until we reach 37 degrees Celsius. So now we are in green. Should we still be vasodilating? That's why we stop. You stop now because you already arrived at the destination. When you're doing the Google map and you arrived at your home, do you stop or do you keep going into the garage? So you gotta stop. That's why some people have a disease. They cannot stop. So this gonna keep on and on. What happens when the vasodilation goes on and on and on? Now we're gonna go from 45 down to 37, and now what's gonna happen now? It's going to go down even more. So 30, 25. So now we have hypothermia. So if you don't stop, you're going to switch from hyper into hypo, which is still stress. So you must be able, people forget about the last stop, the termination, the stop. You're going to have to stop when you arrive at the green homeostasis. I mentioned to you one effector, the blood vessel. That's not the only one. There's two of them. What is the second one? When you're so hot, what happens? Do you just your blood vasodilate at the surface of your skin? What else happens when you're hot? You sweat. Very good. So the second effector is the sweat glands. So the sweat glands are going to receive stimulation, secrete sweat. What is so special about sweat? Sweat has a lot of water, and water is the solution of life. It has very important properties. Water is a versatile solvent. It dissolves most of substance. Obviously, water does not dissolve lipid. Water does not dissolve fats. Water does not dissolve detergent. That's why when you put soap in water, it don't dissolve. But water dissolves majority of things. If there's a mysterious solute that needs to be dissolved, who's more likely to dissolve it than anybody else? It's water. It's a versatile solvent. That's one property. The second property is water has very high specific heat capacity. So water can absorb so much heat. So when you are so hot, what happens is the water is like a sunscreen. The water is a sunscreen. So people put lotion on their face. That's the second sunscreen. The actual first sunscreen is the sweat. It's all over your body. It encapsulates you. It's like a guard. So now it will say, the sweat will say to the sun, hey sun, you're not going to get to the body. You're going to have to get through me first. Over my dead body. That's exactly what happened. The sweat is over there, and the sweat is taking so much heat and evaporating on your behalf to prevent heat from getting into your body. And also, the sweat here is also because sweat is colder than your hot body. It helps absorb more heat. So sweat is working in all directions. It's deflecting the sunlight. It's also absorbing sunlight and evaporating, and on the from below, it absorbing the heat as well. And that what makes you, so that's the effector. The effector was the blood vessel and the sweat glands. And then we reach homeostasis, then we stop. Then we have restored euthermia after we were suffering from hyperthermia. Now let's look at what happens. Let's look at what happens with the opposite. So now we're not dealing with hyperthermia, we're dealing with hypothermia. We're too cold now. What happens in hypothermia is, who's gonna be the sensor? Who's gonna detect the stretch? I'm sorry. Who's gonna detect the change in the temperature? I'm sorry? Yeah, the thermoreceptors. They're gonna detect there's an increase in temperature or a decrease in temperature. The thermoreceptors, when we have hypothermia, they're gonna detect an increase in temperature or a decrease in temperature. Decrease in temperature. Then the decrease in temperature is going to be sent as an input sensory signal, and the input sensory signal will tell the integration center. In this case, because we're still dealing with hypothermia and hyperthermia, is going to be the hypothalamus, and the hypothalamus will understand we have a drop in the temperature. We need to repair it with negative feedback by increasing temperature. Before, when it was too hot, it integrated that there was an increase in temperature. We repair it with feedback to drop in temperature. But now it's going to decrease. It's going to see that there's a drop in temperature. So the output motor signal is going to be the opposite. It sensed there is a drop in temperature. The output is going to be to increase temperature. So now, are the effectors going to be the same? The first one, blood vessel, yes. So now blood vessels, when we were too hot, they vasodilated to allow for heat to leave the body. When we're too cold, do you think they're going to vasodilate to allow the heat to leave the body so we can get even more colder? Or do you think they're going to vasoconstrict so that they can prevent the loss of heat? What do you think they're going to do? They're going to vasoconstrict. They're going to vasoconstrict. They're going to vasoconstrict. When they vasoconstrict, they preserve the heat. That's the first effector. Who's the second effector? Sweat glands? No, it's the skeletal muscles. So the skeletal muscles are going to start to shiver. You're going to start shivering. Energy moves from one form to the other. In your car, you put gasoline. That's chemical energy. When you start the car, there's a combustion, explosion. So now the gasoline is going to blow up inside the car to give us heat energy. So now we shifted chemical energy into heat energy. Then the heat energy moved the piston in the transmission of the car. So now you have mechanical energy and the wheels of the car start rotating. So here we saw that energy shift back and forth. Now what happened here? What happened here is that we shift energy also. So your hypothalamus will send action potentials, electrical energy, and the electrical energy is going to be converted into skeletal muscle energy. So now you're going to start shivering. So this skeletal muscle mechanical energy is going to be changed into ATP. And ATP, that's energy. So that's chemical energy. And you're going to use that chemical ATP like gasoline, like a Bunsen burner to warm yourself and become more warm. And then we're going to become more warm and we're going to restore homeostasis. And after we restore homeostasis, we stop. We stop vasoconstricting. We stop shivering. Otherwise, we're going to go now up into hyperthermia. Then we terminate. Okay? So here we see that as time went on, and this is the temperature. This is temperature. When temperature went up, when temperature went up to 45 degrees, what happened? What happened was there was vasodilation, and there was sweat gland stimulation. But when we get cold, what happened was there was vasoconstriction, and there was also, when we were cold, what happened? There was vasoconstriction and shivering. Excellent, shivering. It's called shivering thermogenesis. Shivering thermogenesis. Shivering genesis means making. So shivering thermogenesis. We shiver to genesis or make thermal heat. So we shiver to make heat. Or they call it shivering thermogenesis. So now what we did here is two variables. And that is, how do we restore thermal? How do we restore thermal energy? How do we restore thermal energy? Is this the only variable we deal with? Now let's look at sugar, blood sugar. We're supposed to have euglycemia or glycemia means sugar. Glycemia means sugar. In medicine, we have our own language, our own terms. So now, if I need to have homeostasis for sugar, what is that called? Euglycemia. Euglycemia. Okay. If my sugar goes up really high, what is that called? Hyperglycemia or hypoglycemia? Excellent. That's hyperglycemia. Hyperglycemia. Now let's look about how do we repair hyperglycemia. What's the first step? Sensor. Who's going to sense that we are not at euglycemia, we have extra sugar? You think a mechanoreceptor, a stretch receptor is going to sense that? Or do you think a thermoreceptor is going to sense that? Or do you think a chemoreceptor is going to sense that? Or do you think a nociceptor is going to sense that? A chemoreceptor. Okay? So a chemoreceptor is like taste. It tastes. There's extra sugar. Too little sugar. Extra salt. Extra calcium. That's the chemoreceptor does that. Tasting. So the chemoreceptor is going to say there is hyperglycemia. It's going to send an input sensory signal to the integrator, to the computer. Who's the integrator now? The hypothalamus? No. The pancreas. The pancreas. So the pancreas is going to feel that there is an increase. It's going to feel there is an increase in blood sugar. So is it going to do negative feedback or positive feedback to repair the problem? Because we have high glucose. How is the pancreas going to integrate the information? We have high glucose. Should we drop it down with negative feedback, or should we increase it even more with positive feedback? Negative feedback. So it's going to initiate negative feedback by sending an outer motor signal to the effector. The effector now is the pancreas as well. So sometimes you can have the same integrator and the same effector. So the pancreas integrated, and it caused the secretion of insulin. So now we're not dealing with vasoconstriction. We're not dealing with sweat gland. We're not dealing with shivering. That's a whole new system. That's a whole new variable. So now the effector here is going to be the pancreas. Then the pancreas is going to send insulin. This is the cell membrane. That's inside the cell, and that's in the blood. Where is the hyperglycemia? Is there so much sugar in the blood, or is there so much sugar in the cell? When we say hyperglycemia, what do we mean? Do we mean that there's a lot of sugar in the blood or a lot of sugar in the cell? In the blood. So what insulin is going to do is going to pluck cells, is going to pluck channels known as GLUT. G-L-U-T. What's GLUT? Glucose transporter. Why? Because glucose cannot penetrate the cell membrane. The cell membrane is lipid. It cannot cross it. So it's going to need a facilitator. It's going to need a carrier, a transporter. So Insulin is going to plug in so many transporters for glucose, so many transporters. So now what's going to happen is glucose is going to diffuse down concentration gradient from where there is so much blood, where there's so much sugar in the blood, to where there is a little bit of sugar in the cell. So now sugar coming in, sugar coming in. So when sugar is leaving the blood, what happens to the hyperglycemia? When sugar is leaving the blood, what happens to hyperglycemia? Does it go down or does it go even more up? It goes down. So now we go down to homeostasis, and we have come down from hyperglycemia into euglycemia. And then what do? We stop. Because imagine you keep more insulin, more insulin. So now we're going to go from hyperglycemia to euglycemia to hypoglycemia. So that's why we stop, we terminate. So that's the loop for hyperglycemia. For repairing, that's the negative feedback loop for repairing hyperglycemia. For hypoglycemia, now let's look at what happens in hypoglycemia. In hypoglycemia, so what's step number one? Sensor. Who senses that there is a drop in sugar? Is it a mechanoreceptor, stretch receptor? Is it a thermoreceptor? Is it a chemoreceptor? Is it a nociceptor? Or is it a nociceptor? What is it? Chemoreceptor. It's going to detect there's a little bit of sugar. So now it's going to send input sensory signal to the integrator, who is still the pancreas, telling the pancreas what? Telling the pancreas there is a lot of sugar or there's a little bit of sugar. Little bit of sugar. It's going to tell it there is hypoglycemia. And you need to repair the hypoglycemia with the opposite, by increasing blood sugar. Do we repair the integration with positive feedback or negative feedback? Negative feedback. So the output motor signal is going to go to the effector. It's going to go to the effector with the pancreas. And now are we going to secrete insulin again? No, we're going to secrete glucagon. Glucagon. Glucagon is going to go into the liver that looks like bow and arrow. You know the liver, when you have the bow and arrow like this, you pull the arrow. So the liver, it looks like that, and then it's gonna squeeze it to burst out a lot of glucose. Glucose is stored in the liver as belt of many glucose known as glycogen. Okay? Now this glycogen is gonna burst out so much sugar. So now what's gonna happen? There is a lot of sugar in the liver. This is the liver. It has a lot of sugar. The blood has a little bit of sugar. Where do you think sugar is gonna come from? Is it gonna move from the liver where there is so much glycogen into the blood? Or do you think it's gonna move from the blood into the liver? From the liver to the blood, because we have hypoglycemia, right? What are we repairing? We're repairing hypoglycemia, a drop in blood sugar. So now, before when we had hyperglycemia, we had a lot of sugar in the blood. We took it inside the cell. Now it's the opposite. Now we're going to remove sugar from the cells of the liver into the blood. So now what's going to happen to the hypoglycemia? Is it going to go up or is it going to go down? It's going to go up. So it's going to go up. So who was the effector here? My liver. My liver was my effector, because it is the one who received glycogen, who has glycogen, and when glucagon was secreted, it was able to burst out all of this glucose, and then we go up from hypo to euglycemia, and do we keep going on and on and on? What do we do now? We stop. So that's another system now. So that's another system. We had a system with thermia, and we had a system with glycemia. And it can go on and on. With the hypertension, your hypertension, when the blood pressure, when you wake up in the morning, what happens? You are laying down. You wake up in the morning to stand up, and the blood is going to fall from your head and neck down to gravity. So now, do you think that's hypotension or hypertension? See, hypo, because the blood dropped, right? You were sleeping, then you stood, then the blood is falling from your head and neck down. So now your brain doesn't have enough blood. There is hypotension in the blood. So in the brain. So now what happens is, who's gonna detect that there is hypotension? A mechano stretch receptor? Who's gonna detect that the blood pressure went down? Is it a mechanoreceptor or a stretch receptor? Is it a thermoreceptor? It's not a thermoreceptor. Is it a chemoreceptor? Not a chemoreceptor. What do you think is a chemoreceptor? Are we dealing with a chemical? Are we dealing with glucose? Are we dealing with calcium, potassium, sodium? So blood pressure? No. Who said yeah? No. What is tension? Tension is blood pressure. When the blood pressure is going so fast, hitting the walls of your blood vessels. So the blood is coming out of the heart. This is the heart, and the blood is coming out so fast, hitting the walls of the blood vessels. So how can that be detected by chemoreceptors? So it's not chemoreceptor. Is this pain? Do you have pain with for the hypotension? No, the pain is a later stage when you have a stroke or when you have like a migraine. So who detects that there is blood pressure? What is pressure? Mechanoreceptors. So how do the mechanoreceptors detect there's pressure? The blood vessels are dilating because there's so much blood flowing and hitting their walls. So this is the blood vessel. This is the blood vessel. This is the blood vessel. And blood is striking, hitting the walls. It's going so fast, turbulent, hitting the walls. So there are stretch receptors sensing, oh, blood pressure is going is so high pressure, hypertension. And the opposite, when blood is not hitting the walls hard enough, they're gonna say, hey, if you come to school on the weekend and the guards look around, there's not a lot of students. There's not a lot. They can see, they can tell. So here, the stretch receptors will say, we're not being stretched. There's not a lot of students. Or here, if you have like 1,000 students at one time, then that's they're gonna say that's too much. But if you have like 10 students, you're gonna be, hey, that's too little. So the stretch receptor is the same. They're like, we're not being stretched as we should be. If they are too stretched, they will say there's hypertension. If they are minimally stretched, they're gonna say that is hypotension. So who senses pressure changes? It is always the mechano or stretch receptors, like the baby pushing the cervix. That's pressure. That's pressure. Or when the baby's suckling the milk, that's pressure as well. So now the step number one, the sensor, stretch receptor, an input sensory signal is gonna go into the integrator, who happens to be your hypothalamus now. It's not the pancreas anymore because we're not dealing with sugar. We're dealing with hypertension. So that's hypothalamus. So the hypothalamus will see that there is a you woke up and the blood fell. There's a decrease. So there's a decrease. They will see there is a decrease in blood pressure. And do we repair a decrease in blood pressure with a negative feedback loop or a positive feedback? Do we repair a decrease hypotension? Do we repair hypotension with a negative feedback or a positive feedback loop? Negative feedback. So that means we need to increase the pressure now. We went to hypotension, we need to come back up. So now the output motor signal is going to be increase the pressure. When the pressure has increased, the effector here is not going to be my pancreas anymore because I'm not dealing with sugar. Who's going to be my effector now? My heart. So my heart is going to increase the heart rate. My heart is going to increase the blood pressure. My heart is going to increase contractility, and now blood is going to start flowing back to your brain and perfusion. What's perfusion? Perfusion is when you give the tissue the blood. Why is blood important? Because blood has oxygen, because it has nutrients for ATP and for nutrition, and because blood also removes all the metabolic waste that builds up and the carbon dioxide. And then when we have the proper tension, we move up from hypotension to eutension. We are at homeostasis. Then what do? We stop. And then we have restored homeostasis. The special name of the mechanoreceptors and the stretch receptors that deal with blood pressure, they have a special name. It's called baroreceptor. 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 I made a mistake here. The integrating center is not the hypothalamus. It's called the brain stem. Brain stem. It has a center inside the brain stem known as the cardiac center of the brain stem. So now we talked about the cardiac center of the brainstem, and now we have seen how you see the theme is happening over and over again. You see that the theme is happening. We just need different variables. That's how homeostasis is restored in your body by these steps. Maybe the players are different. Maybe the integrator for one of them is the pancreas. The integrator for the other one is the brainstem. The integrator for the other one is the hypothalamus. But in the end, you must go through these stages. So you really must know that. You must know that that's how. So later on, when you do medications, you will see that these medications do their actions here. They interfere with these, or they help, they augment these steps to help you restore homeostasis. Okay, so that's the branch of of anatomy and physiology that we're going to be doing this semester. Is this all the anatomy and physiology? No. We said that we begin with the cell and we move up. Cell, tissue, organ system, cell, tissue, organ system, organism, then there is a population. So now we study with reproduction. So when we study the male reproductive system and the sperms and the female reproductive system and the ovary and the eggs and the growth and development, over there, we're not going to be talking about that. We're not going to be talking about sensor homeostasis, because now we're not talking about the health of the person, the individual. Now we're talking about the entire population. That doesn't mean the people do not have sicknesses in their reproductive systems, like men have benign prostatic hyperplasia, and women have cystic, polycystic ovary disease. These are separate diseases, but we will get to that. But I need you for now to know that in biology and in the anatomy and physiology, we are concerned with homeostasis, and we are concerned with reproduction. We are concerned with reproduction. In the PowerPoint slides, when we do the PowerPoint slides, we're going to see that we're going to begin with an understanding of how science developed. So science today is not what it used to be back in the day. Science has developed a lot. Back in the day, they used to do science on slaves. They used to do science on prisoners. Then they realized this is unethical. I mean, they should not be subjected to these kinds of unethical things. So they banned research on human beings altogether. Then they realized, okay, maybe we can do it on dead bodies, cadavers. Then they were having superstition. Said, disease, you are sick because the demons and the gods of this planet and that planet are not happy with you. You need to make sacrifice, all that stuff. But some very important scientists like Hippocrates said that, okay, we should restrict science to the scientific method. We should have hypothesis. We should have hypothesis that is testable hypothesis. We should have falsifiable hypothesis that we can prove wrong, hypothesis that we can reverse, and we can test. So we can test, and then we should have a control group. So if I have a medication and I want to test its efficiency, I want to test the effects of this medication on lowering the pain, the nociception, bringing down the pain sensation. And I name my medication Tylenol. So I should give a group of 50 people Tylenol, and a group of another 50 people, give them a capsule full of water. and don't tell them that it doesn't have Tylenol, and tell both groups to take it to find out if it's effective or not. So if the pain went down in the group who took the Tylenol, that shows you the medication is effective. But if you never had a control group, you can never conclude that the pain went down because of your medication. It could be something else. But when you had another group who never took the medication and their pain never went down, now that really shows it was your medication who brought the the pain down. Then there is the main thing in the lecture before we now go and do the lab. The main thing is that we must know our hierarchy. We must know that we begin from. Did everybody get this? These steps? Okay. 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 Remember I said I'm recording. I'm gonna post it for you guys. Okay. You wanna have two copies? Okay. All right. Okay, guys. Okay. So we begin our hierarchy with the concept that cells are the building block of health and disease. We said that we are healthy because our cells are healthy, and we are sick because our cells are sick. So we move on from cells, and many cells together they give us tissue, and many tissues together they give us organs, and many organs give us organ. Many organs give us organism. Before organism, organs meaning I have a epithelial tissue, I have a liver, so organ, a liver, a heart. So many organs together give us a organ system. When they all working together, then many organ systems give us an organism, and many organisms give us a population. Very good, population. So now they started to learn that beneath the cell there is a very important foundation, and that is we have atoms, we have molecules, we have enzymes, we have organelles like the endoplasmic reticulum, like the mitochondria, like the cytoplasm, and so on and so forth. Cytoplasm. Okay, they learned that back in the day, disease was only seen at the sign of the organ. So if your organ is wrong, you're coughing, your lung is coughing, or maybe you have some tumor. So they thought that the problem is at the level of the organ. Then they realized that's not true. The problem began from the atom, and it went up. So if I want to repair the problem, I must repair from the cell and downward. So even though these compartments on their own they are useless, but together, when they work together, they give me a functioning cell. So for example, when it comes to cancer is when we have uncontrollable cell division. So the cell cycle in cancer is going on and on and on and on, and you have mitosis, all the time. The reason is because you have molecules and you have enzymes and proteins that work as tumor suppressors. Their job is to suppress and stop this aberrant growth, stop this repeating on and on and on, this repeating uncontrolled cell division. But when there is a defect, a mutation that destroys or breaks these molecules, then you're going to end up with a diseased cell. When you have a diseased cell, then you're going to have a diseased tissue. Then you're going to have a diseased organ, then diseased organ system, and so on and so forth. So you must repair the problem at the level of the molecule. How can I